Hello, everyone. My name is Will Fett, and I'm with the Iowa Agriculture Literacy Foundation. Welcome to our STEM Festival. I'm excited to share a DNA extraction experiment with you today. And for that, you're going to need a couple of different household items. If I was doing this in a lab, I might use some different materials like test tubes and beakers, but since we're doing this at home, everything that you'll see here can be found mostly in your kitchen. So first off, we are going to need some salt. We're going to need some regular dish soap. Really, any brand, any kind is fine. Um, I'm also going to use some alcohol. Now, this is rubbing alcohol. It's a 70% alcohol, but any clear alcohol will work. If you use a lower proof alcohol, maybe vodka, make sure to ask your parents. For this experiment, you might want to chill the alcohol because a colder temperature will work better if it's a lower proof alcohol. But this is rubbing alcohol. You can use it to clean things. I've got a strainer. I've got a blender. I've got some water, a spoon, and then a roll of paper towel to clean up if there's any mess here. So we're going to do DNA extraction on soybeans. And you might have seen this experiment on things like strawberries or spinach. Really, any organic matter will work for this. Strawberries and spinach work really well because you can get a lot of DNA and it's very visible. But the reality is that we don't grow strawberries, a lot of strawberries here in Iowa. So I wanted to do something that we do grow a lot of, and that's soybeans. Now, if you think about research and why we might extract DNA and investigate DNA, you need to think about the benefits to the crop. So this is a soybean seed, and this little tiny seed is going to turn into a plant that's going to produce a lot of different pods, and each pod is going to have 3 to 4 more seeds in this. Now, soybeans, if you harvest them green, are called edamame, and we can eat those raw, and you'll see them in a lot of Asian dishes. You certainly can see them in American cuisine as well, but it's not quite as prevalent. But these little dried beans are left on the plant until the plant is completely dry, and these beans can be harvested and made into animal feed, can be made into soybean oil. Vegetable oil is largely made from soybeans, and a lot of other products, industrial products, food products. This little bean has a lot of potential. Now, when we do research, we're looking at the DNA characteristics that will make this plant grow better, stronger, healthier. We might take a little snippet of DNA and investigate how to make the plant drought resistant. So if we don't get enough rainfall, that the plant won't wither and die. Now, DNA is the building block of all life, so we really want to study it and understand the potential that's in that DNA. So this is our little soybean seed, and I've got quite a few here, but dried, they're really hard. So what I did is I soaked them overnight, and that round bean actually, or it kind of looks like a pea to start off with, it actually plumps up and becomes a nice bean shape. And so these I soaked overnight. They absorbed a whole bunch of water, and that's going to help soften them up. Step one, we want to get these into much, much smaller pieces. So I'm going to blend these, and I'm just going to pour them all in with the water. And I'm going to actually add a little bit of extra water because I want to make sure and get a nice liquid. You want to get them into as small pieces as possible because what we're trying to do is get down to DNA. Now, most times DNA is not visible to the naked eye, but this process allows us to get it into small enough pieces and then separate it out so we can actually visibly see it. We won't be able to see the individual codes of the DNA, but we can actually see what DNA looks like with just our naked eye. So this bit's going to get a little bit loud. I'm going to turn on the blender and let it run for about a minute. So if I were to let it run a little bit longer, I think I'd actually get better results, but I want to kind of keep moving forward with the experiment here. Next step, I want to take our strainer and I'm trying to get the liquid off. So hopefully there's been some pulverized plant material mixed in our liquid, and I'm just going to strain off some of that liquid. Now, obviously there's a lot of genetic material in this blender. I don't need all of that. I can certainly go back and try it again later, but I kind of just want to get some of the liquid. Really, we don't need much because DNA, there's so much DNA, all living matter is made up of DNA that we don't need a whole lot to work with here. I'm going to go ahead and let that drain a little bit. And ultimately, we're trying to get down to the smallest building blocks of life. So I don't have much liquid here, but there is DNA in that liquid. Next step is going to be to add some dish soap. What this is going to do is help break down that organic material. And specifically, we're trying to get rid of the fat molecules. So the fat molecules are going to bind to that soap. I'm just going to give it a little swirl to try and mix in that soap. I want to try to avoid air bubbles as much as possible in this experiment. That just complicates things a little bit. So I've got my soap in, and then the next thing I want to do is add a little bit of salt. So salt is going to act like an enzyme and start unraveling that DNA from the protein structures. So I'm going to add just a pinch of salt, maybe a little bit more for good measure, and go ahead and give that a swirl. Now, if I was really wanting to get good results, I'd probably just let that sit, let the salt do its work, let the soap do its work, and allow the DNA to become separate from the fat, separate from the protein. But I don't have that much time, so we're going to move on with the next step. The next step is to insert our rubbing alcohol, our isopropyl alcohol. Now, alcohol is insoluble, so I want it to sit on top of the liquid. If you have a pipette, use that. We're just going to run the alcohol down the side of the glass so that it doesn't mix with the liquid, but it just kind of sits on top of the liquid. But who has a pipette at home? So you can actually do the same thing very carefully. We're going to put the liquid alcohol right on top, let it run down the side of the glass so that the two solutions stay separate. Now, the longer I let this sit, the better my results are going to be, but you can start to see those little white floaties in the liquid. That's the DNA. So pretty cool that within a matter of minutes, we have broken down plant material, we have separated the DNA from the protein and the fat molecules, and now that's starting to rise up into our alcohol solution. I have one I did just a little bit ago, and you can start to see that it's settled out a little bit more. All of those white floaties, that's the DNA. Now, if we had a really high-powered microscope, we could start to look at those and start to really understand the genetic makeup of that soybean and how we might be able to use that genetic makeup to improve the plant and ultimately improve the yield that that plant is going to give us. So you can do this experiment at home. I encourage you to try whatever kind of plant matter that you have. Bananas work great, strawberries, spinach, but I'd also challenge you to try an Iowa crop and see what kind of genetic material and DNA you can extract. Good luck, have fun.